Hi Neuroscience Club! This is the first video of our new club program, a series of recorded lectures on the BrainFacts book to help everyone learn more about the brain and prepare for the brain bee. This video is going to cover Chapter 1, Brain Basics, and I'll be going over the basic anatomy of the brain as well as key concepts of neural networks. So the general idea is that humans are able to do all these actions, as you can see here, simultaneously breathing, jumping, forming memories, communicating with friends, because of your brain. Your brain is controlling these actions both voluntarily and involuntarily, and it is able to multitask because the brain has multiple areas, each dedicated to specific functions. So now let's look at these areas or at least get a basic idea of the divisions of the brain using this diagram right here. So the largest part of the brain is the cerebellum, which is this area that I'm shading right here. So all of this is what we would call the cerebellum. And notice that in this view of the brain, the eyes are on the left side, so right here. And this is what would be the left view of your brain, because your cerebellum is divided into two hemispheres, the left and the right hemispheres. These hemispheres are connected by nerve fibers, which we'll describe more later, but they're basically for carrying information from one side of the brain to the other. And the main nerve fiber that does this is called the corpus callosum. So that's an important term for the brain bee, which is the main nerve fiber that connects the left and the right hemispheres. And it's a bit hard to see the corpus callosum in this view of the brain, but if you go to the sagittal view of the brain, which is basically if you were to cut the brain in half from top to bottom, which we'll be doing in our upcoming BrainFacts section, so we should set up for that, you will be able to see the corpus callosum. So again, the BrainFacts book doesn't actually talk about this, but it is commonly tested in the brain bee. So the corpus callosum in this diagram is this long region right here. That is the corpus callosum. Now going back to this previous diagram, the cerebellum is covered by a layer of tissue called the cerebral cortex. It's a bit hard to identify, but it's basically this tissue that's covering this entire region. It's deeply folded, hence all these marks, these folds, as you can see here. And the basic concept in biology is that whenever something is deeply folded like this, it's to increase surface area, which allows more information to come in and more material to pass through, generally making the entire system more efficient. So again, that's the cerebral cortex. This deeply folded layer of tissue. And as you can see, there's some main divisions shown in this diagram here. The entire thing is the cerebellum, we can cover later, but we can divide the cerebellum into four main lobes. The frontal lobe, which is for your higher cognitive functions and some voluntary movements, personality, things like that. Your parietal lobe, which is mainly for integrating visual signals. Your occipital lobe, which is in the back of this brain, it's kind of small over here, which is mainly for integrating visual signals. So parietal is sensory in general, and occipital is mainly for visual. And your temporal lobe, which is quite important, it's down here, and it has multiple functions, but probably most importantly, as the BrainFacts mentions in this area, in this part of the chapter, it contains the hippocampus and the amygdala. So the hippocampus is mostly for forming new memories, which is different from storing memories, that's important to note, and the amygdala is for fear, motivation, rewards, emotion, things like that. And they're both part of the limbic system, which is just a system, obviously, a structure, a collection of structures that regulate emotion. So it's a bit hard to see the hippocampus and the amygdala on this type of diagram, but it's really important that you know how to identify them on the test. So if we turn to a diagram of the limbic system, specifically the limbic system, you can see the hippocampus and the amygdala. They're in these regions. They're pretty close to each other, but you have to approximate the location on the test, and they're all in the temporal lobe. And the limbic system also has two more important structures. There's many more, but in the book it mentions two more in this part. The thalamus, which is not labeled here, but it's like this circular region over here. That is the thalamus, and its main function in regards to the nervous system is to receive sensory signals, integrate them, and then send them to the appropriate location in the brain. There's also the hypothalamus. Which sends hormones through the pituitary gland. And we'll be talking more about this in future videos, so look forward to that. And here, if you remember, we also talked about three main divisions of the brain: the forebrain, the midbrain, and the hindbrain. So the forebrain is basically everything that we talked about so far. That's the cerebrum, the cerebral cortex that covers the cerebrum, and most parts of the limbic system. So that's the forebrain, and it's quite big. It's all this region right here. And there's also the midbrain. The midbrain is a bunch of structures, and they're mainly for involuntarily movement. So it's a collection of structures that regulate involuntary movement, the most important of which is the basal ganglia. The basal ganglia is always hard to locate because it's sort of inside the brain, and you would have to cut the brain open and look at it from the bottom to be able to see it. And lastly, there's also the hindbrain, which is made of the cerebellum, which is different from the cerebrum. It's important to note. And the pons and the medulla. The cerebellum is this shell-ish region. It's also a bit circular over here. And as you can see, it's the second largest area of the brain, and it's mainly for voluntary movements. It controls your voluntary movements. The pons and the medulla, this is getting a bit cramped. Let's go back to this diagram previously. The pons is sort of the circular region over here. The pons, and your medulla is sort of like this. I should probably use highlight. So I'll highlight it blue. This region, or medulla oblongata, they mean the same thing. Pons, the functions are breathing and posture. Well, there's many functions, but these are the two main functions. And your medulla is for basic functions. A bunch of basic things that you can't live without: breathing, your heart rate, things like that. And also it connects the brain, which is obviously up here, to the spinal cord. That's the spinal cord. And if you notice in this diagram, the midbrain is sort of around here. So if you add all that together, so that's the purple, which is the midbrain, and the green, which is the pons, and the medulla, which is the blue over here, then you have your brainstem. So that's where the term comes from. Brainstem. It's quite important. Now, those are the main areas of the brain, but these areas don't work alone. They have to be able to communicate with each other. This is done through neural networks. It's quite a complicated concept, which we'll talk about later. But first, we have to look at the smallest unit of communication. Your brain, of course, is made of cells, and these cells are called neurons. So we find drawing of a neuron somewhere. Here we go. This is basic diagram, basic structure of the neuron. You have the cell body, otherwise called the soma, the dendrites, which are these little protuberances, and they basically receive signals. So if there would be another neuron over here, then it would receive, dendrites would receive signals from this neuron. And the axons. Axons is a long line that goes in some direction. And if we were to look at a resting neuron, which is when the neuron isn't receiving an impulse, a signal, and it's not sending a signal either. So if you're like to look at the axon of a regular neuron, so pretend this is the axon, there is a constant flow of ions, which are just charged molecules, going in and out of the cell. So there's these ion channels, ion channel, and ions going in and out. And in a resting neuron, there are more positive ions going out of the cell than there are going in. So more positive on the outside than the inside, then that's where you have what scientists like to draw as this. That is called the membrane potential, which is just like the charge, I guess. So in a resting neuron, the charge is negative 70 millivolts, which means it's more negative on the inside. So the inside of the neuron is more negative, that's where you get the negative number. And that's in a resting neuron. But however, if there's an external stimulus, so I guess the first step to neurons sending signals is for there to be an external stimulus, something that happens on the outside of the cell. External stimulus is what they basically do is they open some sodium channels. So open Na plus channels, allowing sodium to come in. So again, we have this axon, and normally it's plus, 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 minus, 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 minus. But if we have an external stimulus, and there's a sodium channel over here allowing sodium ions inside the cell, well, sodium ions are positive, right? Right here. So if we have more positive ions coming in, then now this region is going to have a positive charge. And if this region becomes more positive relative to the area outside of it, then its membrane potential, which we mentioned earlier, will rise. Over here, membrane potential rises. And if it rises to a point where it passes what we call the threshold voltage, which is around negative 55 MV, because remember originally it was negative 70. If sodium comes in, it becomes more positive, it increases. If it passes negative 55, then all the sodium channels in the region will open and sodium will rush in. So rises if negative 70, then Na plus channels open and sodium comes in. So if I were to draw that, what would happen is in this area where sodium comes in, now the charge is positive. The outside is negative. But over here, it's still negative because the sodium is only coming in this area. However, eventually, if this continues, means sodium keeps on coming in because the external stimulus is just so strong, then there will be a domino reaction of positive charges down the axon. So what I mean is these sodium will kind of move over there, right? It'll flow to the right, and what will happen, something looks like this. And over here it becomes back to negative. So this is the area that's positive. And again, if the external stimulus is really strong, then the positive chain will continue going down. So that is what we call the action potential, which is basically just domino reaction of positive charges down the axon. And this travels all the way down the axon until you reach the axon terminal. Again, this phenomenon is called the action potential. Again, if we were to go back to this diagram, the axon, you have the action potential going this way until it reaches the axon terminal, which is just this area, the terminal, axon terminal. And the action potential gets passed on to the synapse. So I guess you could call this entire region what you would call a synapse, which is just the junction between neurons. So if we were to look at the synapse and what happens there in more detail, you can look at this diagram over here. It may seem complicated, but it's not that complex. Actually, it's complex, but it's not that bad. And remember, this all happens really fast. So the action potential is passed on to the synapse. So action potential reaches presynaptic terminal. And what basically happens is that these positive charges causes calcium channels to open, allowing calcium into the cell. So calcium is inside the cell. And what they do is they basically bind to neurotransmitters which are just signaling molecules for neurons. And these neurotransmitters are released from the axon terminal and drift to the next neuron. So it drifts in these vesicles to the end, the neuron, through the synaptic cleft, which just drifts to the next cell because the next cell is over here, and it binds to receptors on this next cell. So these postsynaptic channel receptors binds to receptors. That's the external stimulus for this next cell, and it repeats the cycle. Now here it's important to note that the effect of these neurotransmitters binding to these dendrites of the next cell can either depolarize the neuron, write that down, can either depolarize, which means it makes it less negative. And remember, the threshold potential, if it's less negative, it's more positive, so it's more likely to meet that threshold voltage, and it causes it to continue firing action potential. So the action potential continues, the signal continues, the message continues to get sent. Or it can hyperpolarize, which is the opposite. It makes it more negative, so it's less likely to meet that threshold voltage. So now the action potential will stop. So the action potential stops. And from these two effects, we can get two classes of neurons. Neurons that depolarize are excitatory neurons. And the most common excitatory neuron is glutamate. This is very commonly tested, so make sure you know that. And neurons that hyperpolarize, obviously, are inhibitory neurons. And the most common type of inhibitory neuron is GABA. Again, so now besides these two main types of neurons or types of neurotransmitters, there's also two main types of postsynaptic receptors. So those are these receptors that, remember, the neurotransmitters were binding to when they reached the second neuron. So there's two types of receptors. There's metatropic receptors and ionotropic receptors. So the main difference is how the ion channels are being affected by the neurotransmitter. So I guess in the simpler one, autotrophic receptor is if the neurotransmitter binds directly to the ion channel. So NT, neurotransmitter, binds directly to the ion channel. So all it's doing is when the neurotransmitter crosses the synaptic cleft and reaches the second neuron, it just binds to the receptor. So in this diagram, these would be the neurotransmitter that bind to the receptor, the ionotropic receptor, the ion channel opens, and that's the signal. Versus in a metabotropic receptor, the neurotransmitter doesn't directly bind to the ion channel, rather it binds to other molecules that causes a cascade, eventually reaching the ion channels. There's a lot of specifics, including the G protein that's part of the molecules that causes the cascade, but for BrainBeef, that's all you have to know. So that's how neurons produce signals and how they transmit the signals. So that would be how your brain responds to external stimuli. But those are just individual neurons. What about the big picture? Well, earlier we mentioned the term neural networks, right? But first of all, again, we start from the small things. So the first term that the BrainFacts book mentions is nerve tracks. Nerve tracks are chains of neurons that send signals over long distances. So they're like bundles of neurons. So this would be like the corpus callosum that we mentioned earlier. So example, corpus callosum, and the smaller anterior commissure, which connects the left and right temporal lobe. And branching from that, neural networks are groups of nerve tracks that connect regions of the brain. So again, you have those individual neurons that are using action potentials to transmit signals, and then you have these nerve tracks, which are just a bunch of these neurons that send signals over a long distance, and then you have the neural network, which is a bunch of nerve tracks that connect regions of the brain to again send signals. So the book gives the example of processing visual stimuli. So this entire thing, this entire pathway is a neural network. So our next video will be going in detail over how your brain processes senses, one of which being visual signals, but basically, this neural network is the system that routes signals through the brain in linear pathways. So for example, one network might be in charge of carrying visual signals from the eyes to area A, and then to area B over here, and then to the occipital lobe back here. So that is the network, the pathway, the circuit. And the third important term that the BrainFacts book mentions is the neural circuit, which are interconnected neurons that turn entering signals into output patterns that can be sent to other parts of the brain. So for this part of the book, for the competition, you only have to understand kind of like the logic behind these pathways. It's not that important to memorize these specific terms or be able to distinguish between them. So yeah, that wraps up our first lecture on the areas of the brain and how they communicate through action potentials and neural networks. Thank you for listening.